Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course Developmental Biology. I am Dr. Priya Goyal, working as Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College of University of Delhi. In this module, we will take up the topics in vitro fertilization, embryonic stem cells, and amniocentesis. This is the syllabus of developmental biology that we are following. We are done with Unit 1, 2, 3, and 4. And in Unit 5, we are done with the topic teratogenesis. So in the present lecture, we will take up the last 3 topics of the syllabus, that is in vitro fertilization, stem cell, that is the embryonic stem cells we are to take, and amniocentesis. So let us begin with the first part of this lecture, that is in vitro fertilization or IVF. IVF is a type of assisted reproductive technology, that is ART. In vitro literally means in glass. It is a Latin word in which in vitro means in glass. In vitro fertilization literally means a fertilization which happens in glass. The glass is used for the material of the glass with which the petri dish in which the fertilization actually happens, because of which this in vitro word has been taken. So in vitro fertilization is defined as a process in which egg cells are fertilized in vitro, that is outside of the body. As you know, normally in mammals, in humans, the fertilization process takes place inside the body of the females. So in vitro fertilization is a process in which fertilization happens outside the body. And this embryo, which is thus formed, is then transferred back to the uterus of the female to hopefully implant and become a pregnancy. Dr. Robert Edwards and Dr. Patrick Steptoe have been credited with the development of first test tube baby, that is Louis Joy Brown in England, who was born on 25th July 1978. Louis Joy Brown was born as a result of natural cycle IVF, and no stimulation was made in development of this test tube. In India, around 68 days later, Dr. Subhash Mukhopadhyay was the scientist who successfully delivered the first test tube baby in India, who was named Kanupriya Agarwal and commonly known as Durga, who was born on October 3, 1978 in Kolkata. However, this discovery remained largely unknown to many people around the world, and Dr. Subhash Mukhopadhyay also died a few years later. Used for babies produced by IVF is test tube babies. Though actually the babies are not produced in test tube, it's only the fertilization step that is done in a petri dish, and subsequently the successful embryos are transferred into the uterus of the mother, where they complete the rest of their embryonic development. IVF is also not the first step that the infertile couples go for. Initially, the couples are suggested artificial insemination or certain drugs which may enhance the fertility. So, under what causes and what types of infertility a female may be referred in vitro fertilization? Number 1, when the female has infertile tubes. So, infertile tubes literally means when the fallopian tubes may be absent or blocked. This blockage may also be because of some kind of infection, for example, chlamydia infection, can leads to blocked tubes. The second cause of infertility is ovarian infertility, in which the ovaries may be non-functional or may be absent, or in certain cases, premature ovarian failure is also a reason. So during in vitro fertilization, if the ovaries of the female are non-functional or absent, then the donor can be used for donating ovum or egg. The third type of infertility is because of uterus, in which the uterus may be absent altogether, which is known as Mullerian defect, which is congenital in nature. So under such cases, after IVF, a surrogate mother can be used in which the embryo can complete its subsequent embryonic development. The fourth is the cervix specter, which includes the mucus or the pelvic pain during periods, pain during intercourse, heavy fatigue, or painful urination in the female. These are also the reasons for infertility. Endometriosis is a major reason in which the pelvic anatomy is distorted and functional endometrial tissues are generally present outside the uterus. So here you note that normally only inside the uterus is a functional endometrial tissue is present. Endometrium stands for the inner lining of the uterus. Another cause of infertility among females is the hormonal malfunctioning, in which the hormones may be insensitive in their effect, they may be produced in very less amount or even absent altogether. The important hormones that are required in female reproductive cycles is estrogen and FSH. So if there is less estrogen or high FSH, or there are certain autoimmune disorders which may be because of stress mentally, or there can be a physical heavy exercise that a female does. So because of these also the reproductive barriers may arise. The next cause is pelvic cysts, which includes some mechanical disorders. There can be certain age-related factors. The woman's ovarian function diminishes with age. So if the age of the female is quite high, then there are certain chances that the female is not able to conceive as easily. Another reason is PCOS, that is polycystic ovarian syndrome, which includes a large number of disorders which are characterized by irregular ovulation, irregular menses, and obesity and acne, and also weight gain in the females. Or there can be certain unknown idiopathic factors. Now, the types and causes of infertility among males includes the problems with sperm production, with sperm function, or the storage of sperm. The cause of infertility can be azoospermia, which is characterized by absence of sperms. This is also called azoospermia if the number of sperms that are functional is less than 32%. The second type of infertility is oligospermia, in which the sperm count is generally less than 13.5 million per ml. That is, the concentration of sperms is quite low. So under these cases, IVF is helpful. Under normal conditions, under natural conception, the number of sperms required is around 2 into 10 to the power 7, that is 20 million sperms are generally required. However, in IVF, even as low as 10 sperms have been known to cause fertility. Although generally in IVF, 50 to 70,000 sperms are put in a petri dish along with a single, but as low as 10 sperms have also been known to cause IVF because of various variants day by day that have been that are nowadays available. Now, what are the causes of infertility in male? The causes can be genetic, which includes Y chromosome microdeletions. The cause can be a damage to testes, which is an anatomical reason, or it can be an infection, or there can be certain problems with the sperm capacitation, that is, the sperm does not get activated. The reason can be blockage or absence of vas deferens, or the certain lifestyle reasons like tobacco, cocaine, drugs, alcohol, excessive heat, or even excessive exercise, which makes the male infertile. So what are the steps involved in the process of in vitro fertilization? In vitro fertilization, or the IVF, involves hormonally controlling a woman's ovulatory process, then retrieving ovum from the woman's ovaries and fertilizing them with the sperm in a culture medium kept in a petri dish, and transferring the resulting embryos back to her uterus some 2 to 6 days later. This diagram shows the general step-by-step procedure of IVF. In the first step, as you see, eggs are being collected after ovulation or even directly from the follicles. Sperm sample is also collected side by side and concentrated by removal of the seminal fluid. Next step involves fertilization, in which both the collected eggs and the sperms are mixed in a petri dish, and fertilization is allowed to occur. After this, the fertilized zygote is removed from the petri dish so as to be implanted into the uterus of the mother, and this is done by surgical implantation. And after successful implantation, the zygote develops normally. It completes its embryonic development as if it had been normally fertilized in the female's oviducts only. So there is no difference in the development of the zygote, or there are no effects on the baby generally, even if it is fertilized outside or inside the mother's body, because the whole of embryonic development is taking place inside the body of the mother. So now we'll take up the different steps of IVF one by one. The first step involves treatment of female for the procedure, in which the menstrual cycle of the female is first of all selected for recovery of the oocyte. Now, the menstrual cycle of the female can be a natural cycle in which a surge in luteinizing hormone coincides with the time of ovulation. The cycle selected can be a stimulated cycle in which ovulation-inducing hormones are given in the form of a short course, which are predominantly the gonadotropins. They are administered so as to cause development of many Graafian follicles at the same time, so as to draw many oocytes from the female instead of just one. This increases the chances of fertilization. Or it can be a controlled cycle in which the human chorionic gonadotropin is used to arrest the development of Graafian follicles so as to have a controlled ovulation time. Now, the oocyte is retrieved some 34 to 37 hours after the injection of HCG. Along with this, there is regular ovarian monitoring and stimulation done. The monitoring of ovarian stimulation is done by ultrasound, in which the follicle size, even less than 1.9 centimeter diameter, is easily monitored with the ultrasound. The temperature charts are continuously prepared. Any changes in the cervical mucus are noted down. Also, the spontaneous LH surge timing is predicted statistically from the last 6 menstrual cycles of the female, and also the different levels of LH hormone in urine and plasma. This gives the accurate time of ovulation, that is the ovum retrieval during laparoscopy. The second procedure involves oocyte retrieval. Oocytes are generally aspirated at metaphase 2 stage from mature Graafian follicles with the help of a needle directly from the ovary, which is known as transvaginal oocyte retrieval mechanism. And this is done with a laparoscope, as you can see in the photograph here. Now, the tissue covering the ova is scraped away with laparoscopic scissors. The aspirate that contains the oocyte may also contain some blood, some heparinized solution, which is used during aspiration of the oocyte, which is added to avoid the formation of blood clots. Hence, the ova are segregated, the blood and heparinized solution are removed, and the segregation of ova is done on the basis of their morphological characteristics. The best ova are selected and the oocyte culture is done, during which the oocyte are incubated for some 5 to 10 hours. And once the oocyte is mature, it is ready for insemination. Simultaneously, the sperm are also collected from the male partner, and the sperms are collected after a 3-day sexual abstinence, because this ensures the sperm motility and quantity in the sample. The semen sample is collected some 60 to 90 minutes before the procedure of fertilization, that is IVF, and this is collected by masturbation, or the semen sample can be collected from the frozen semen sample. After collection, the semen sample is washed so as to remove any seminal fluid and centrifuged. The sperm pellet so obtained is made to undergo in vitro capacitation and again centrifuged, and further only the capacitated sperm which come in the supernatant are collected and used for the process of in vitro fertilization. Now, the process of in vitro fertilization involves the following steps. The first step is insemination or fertilization, which is done in vitro, that is in the petri dish. During insemination step, the eggs are mixed with minimum 50,000 actively motile sperms, the sperms that have been collected from the supernatant after the step of capacity. Or the fertilization may require a single sperm also, as is the latest variant of IVF, which is known as intracytoplasmic sperm injection. We'll take up this ICSI later on. The second step involves incubation, during which the inseminated or the fertilized egg is incubated at 37 degrees Celsius for some 18 to 21 hours at 5% carbon dioxide levels and 98% relative humidity, which is considered optimum for the incubation. Now, to observe for the success of fertilization, the inseminated eggs are placed under microscope and pronuclei and polar bodies are to be observed. The presence of two pronuclei and two polar bodies implies a successful fertilization. The next step is cleavage. First cleavage occurs after 24 to 30 hours and subsequent cleavage occurs after 10 to 12 hours. Now the uncleaved eggs are rejected and the cleaving eggs are selected for the further process. Further, the embryos are transferred at the cleavage stage into the uterus of the biological mother, or because of any complications with the mother, the uterus of a surrogate mother can also be used for implantation. After the transfer of the embryos into the uterus, they undergo a process known as hatching. We have also discussed the process of hatching during the fertilization lecture. During hatching, the embryo actually breaks off its zona pellucida before implantation, and if generally this happens, but if this does not happen, then an assisted hatching step is added, during which the zona pellucida is partially opened by dissolving the embryo in acid or also with the laser just before transfer, which helps in hatching. That is why this one is assisted hatching step. And the final step is that of implantation, during which the hatched embryo implants itself into the uterus of the mother. Now, apart from the general procedure of IVF, there are certain variants of IVF also, which are done when some change in the conventional IVF procedure is required. For example, the first is intracytoplasmic sperm injection, or the ICSI. This ICSI was started in 1992. The major advantage of intracytoplasmic sperm injection, as the name says, is that number of sperms required is just one. This is helpful in case of male oligospermia or obstructive azoospermia, in which the ejaculatory duct is abnormal or blocked, and thus a large number of sperm are not able to be retrieved for the process of IVF. During this process, the sperms are retrieved directly from the testes or directly from the epididymis, and hence the name testicular sperm extraction, that is TEAS, or percutaneous epididymal sperm aspiration, PESA, or microepididymal sperm aspiration, MESA, respectively, in men which have vasoepididymal obstruction. This is the diagram which shows an intracytoplasmic sperm injection. After the corona radiata and cumulus mass around the oocytes are removed by oocyte processing, a single spermatozoon is directly introduced into the ooplasm, which has been retrieved during IVF, and this process is done with a special needle known as micromanipulator, which pierces through the wall of the egg and is thus able to successfully load a single sperm into the ooplasm so as to ensure fertilization. The next variant of IVF is gamete intrafallopian transfer, or the GIFT. In this procedure, eggs are removed from a woman's ovary and placed in one of the fallopian tubes along with the man's sperm. Oocytes, which are collected laparoscopically, are examined under the microscope for quality, and the best quality eggs are mixed with a washed and prepared sample of the semen of the sperms. The sperm-egg mixture is released into the femoral end of the fallopian tube with a guided catheter. So in this case, the fertilization also happens in the fallopian tube instead of the petri dish, as happens in the conventional IVF procedure. And thus the resulting embryos, which are formed during fertilization in the fallopian tubes, travel down the tubes into the uterus for implantation. So here there is only a change from the normal biological process that oocytes and sperms are collected from the male and the female respectively, and but the fertilization occurs inside the fallopian tubes only. So it mimics the normal process of fertilization in fallopian tube. However, a prerequisite for this process is that female has normal fallopian tubes where the fertilization has to take place and the male is able to produce adequate sperm. A next variant of IVF is zygote intrafallopian transfer or the ZIFT, which combines in vitro fertilization and gift. So during ZIFT, eggs collected during IVF are mixed with sperm to undergo fertilization in vitro. However, the fertilized eggs or zygotes are inserted into the fallopian tubes, not uterus, as in IVF, with the help of a guided catheter, and the zygote travels down the tube to reach the uterus for implantation. So in ZIFT, the fertilization happens in vitro and the zygotes are transferred into the fallopian tube so as to start their fertilization and so as to start their development, unlike the IVF where the fertilized embryos are transferred into the uterus. The next variant of IVF is pronuclear stage tubal transfer, that is PROST. It is similar to ZIFT. It uses an in vitro fertilization, but the fertilized egg is transferred to the fallopian tube at the pronuclear stage. Another variant of IVF is autologous endometrial co-culture, which is a possible treatment for patients who have failed previous IVF attempts or who have poor embryo quality. In autologous endometrial co-culture, the patient's fertilized eggs are placed on top of a layer of cells from the patient's own uterine lining, that is endometrium, so as to create a more natural environment for embryo development. So, in contrast to the conventional process of IVF that was done years ago, there are a large number of variants of IVF that are used these days. There can be certain combinations of these variants which are used for successful pregnancy to help the infertile couple. But it comes with its own complications. A large number of risks are associated with IVF, predominantly the ovarian hyperstimulation syndrome. As we discussed during the procedure, a large number of fertility drugs are given to overstimulate the ovaries so that a number of follicles develop simultaneously. And this process is known as superovulation. But instead of superovulation, the ovaries may respond in such a way that it leads to hyperstimulation of the ovaries, and this is known as ovarian hyperstimulation syndrome. The symptoms of which are that body fluids accumulate in the female abdomen, the ovaries may become enlarged or swollen, and this may affect the vascular, respiratory, or even the renal system of the female that is undergoing IVF. And very rarely, this can be even fatal for the female. The second risk associated with IVF is multiple births. Since more than one embryos are inserted in the uterus of the mother, so as to increase the chances of implantation. So it may lead to implantation of more than one embryos, which leads to a burdening of the uterus. Thus, it causes morbidity or even mortality to the mother, sometimes to the child, or it can even lead to preterm delivery of the child. And if the mother is overburdened, then it leads to some kind of birth defects in the child. The child may be having low body weight, or even it may suffer from some disabilities. The child may suffer from some neurological, cardiovascular, or musculoskeletal defects, like some digits may be missing, or sometimes some extra digits may be present. So instead of implanting more than one embryos during IVF, some extra embryos may be cryopreserved for future courses. Besides this, there are certain psychological aspects associated with IVF. Sometimes the procedure undergoes failure. The female is not able to get pregnant. Thus, it leads to disappointment or depression to the couple who is undergoing such rigorous treatment. And sometimes the couple may have a feeling of being cheated. A large number of money is involved, and thus there can be a guilt of money wastage. apart from the risks involved, there are certain ethical constraints for the process of IVF. IVF bypasses the natural way of conception, and even females who are past the menopausal age can get pregnant. IVF creates a life in lab. Excess embryos which are produced during the fertilization step in the lab, they are discarded. Only a few successful embryos are transferred to the uterus of the mother, while the remaining embryos are discarded. IVF also provides an unnatural environment for the embryos in their initial life. It also has the potential to modify or select embryos, which may be done, which is considered unethical in case of humans. And secondly, it is considered against the law of nature. So these are the ethical considerations against IVF. So this completes the first topic, IVF, and now we will start with the second topic of this lecture, that is amniocentesis. Amniocentesis is also known as the amniotic fluid test. Amniocentesis is a prenatal diagnostic test which is used for detection of genetic disorders and or infections, which is done by using the fetal DNA from the cells which are collected from the amniotic fluid. This is carried out between 14 to 18 weeks of pregnancy. The process was pioneered by American obstetrician Frederick Fish and Danish gastroenterologist Paul Rees in 1956 when it was done for fetal sex determination. However, the first amniocentesis process was done blindly, which means that here the ultrasound techniques were not informed. It was in 1972 that James Bang and Ellen Northland introduced the use of ultrasound techniques. So ultrasound techniques are done simultaneously when the needle is inserted into the amniotic fluid for its connection. It does not allow any kind of harm to the fetus which is growing inside. As you see in this diagram, this is the fetus which is growing in the amniotic sac, and there is all around it an amniotic fluid present. This is the umbilical cord. A syringe is being introduced into the amniotic fluid through the abdomen so as to collect some amniotic fluid into it. And at the same time, the path of this needle is guided with the help of an ultrasound transducer, which is connected to a monitor, which is side by side monitored for the path of the needle that is going around the fetus. So this minimizes any kind of harm or injury to the fetus which is going inside the amniotic sac. A small volume, around 20 to 30 ml of amniotic fluid, is generally aspirated during amniocentesis, and that fluid is then centrifuged to isolate the fetal cells. Actually, the fetus keeps on shedding some of its skin cells and apart from it some proteins and also its urine in the amniotic fluid which is present around it. So to remove those things, the fluid is centrifuged. The fetal cells are then grown in a culture medium and then they are examined for chromosomal abnormalities by karyotyping. So why is amniocentesis done? As it was pioneered initially for fetal sex determination, this is the foremost application of amniocentesis. It is also done for genetic testing, during which the chromosomal analysis is done for detection of any kind of chromosomal abnormalities in the fetus. For example, Down syndrome or Turner syndrome, some kind of genetic disorders, for example, spina bifida, cystic fibrosis, or Tay-Sachs disease. Also, amniocentesis can help in assessing the fetal lung maturity at the time of birth. Generally, the pediatricians are there for assessing the fetal lung maturity of the child at the time of birth. So this amniocentesis can also be done before birth for fetal lung maturity testing in case of any doubts. Amniocentesis is also done for diagnosis of any kind of fetal infection, that is for evaluating the fetus for infection or other illness at the time of birth. It is also helpful in assessment of the severity of anemia in babies who have Rh sensitization. Rh sensitization is a rare condition in which the mother's immune system starts producing antibodies against fetal blood cells. Amniocentesis can also be used to drain excess amniotic fluid from the uterus of the mother in case it is over-accumulated during pregnancy. This is known as polyhydramnios condition. Amniocentesis can also be done for paternity testing, during which the DNA isolated from the fetal cells can be compared to DNA from the potential father. And if the DNA of the two matches, it helps in confirming the paternity of the fetus with its potential father. Each of these blastomeres has the potential of self-renewal or differentiation into any cell of the body. And during this, a cavity blastocele is created during the process of cavitation. The first differentiation event in human occurs at approximately 5 days of development, when the outer layer becomes the trophoectoderm and the inner cell mass gets separated. Now, the inner cell mass will subsequently develop into a cluster of cells called the epiblast that develops into the embryo proper, while a layer of primitive endoderm or the yolk sac cells that together with trophoectoderm gives rise to extraembryonic structures, namely the embryonic site of placenta, chorion and yolk sac. Now, this was the revision of the formation of the embryo in case of humans, as we discussed earlier in the earlier lectures also. So from here now, if the inner cell mass cells are removed from the normal embryonic development that they are supposed to undergo, and then they are cultured under appropriate conditions, they produce embryonic stem cells. Now, these embryonic stem cells are able to retain their pluripotency, and they can generate all cell types of the body as if they are supposed to form the complete embryonic development of the embryo. And in vitro, these embryonic stem cells are harvested from embryos at a developmental stage before the time that implantation would normally occur in the uterus. There are two different pluripotent states of an embryonic stem cell, which is naive and primed. Naive is the one which is most immature and undifferentiated state, which has the greatest potential for pluripotency. For example, most of the existing mouse embryonic stem cell lines. And the primed, as the name says, is the embryonic stem cell which has got some maturation toward the epiblast lineage. So it is already primed or ready for differentiation, as most of the captured human embryonic stem cell lines are. The technique to identify and culture embryonic stem cells was first of all reported by Evans in 1981 for mouse embryonic stem cells, and by James Thompson in 1998 for human embryonic stem cells. Evans got the Nobel Prize in 2007 for his technique of gene targeting using embryonic stem cells. Now, what makes these embryonic stem cells pluripotent? What is essential to maintain a transient pluripotency in these cells is the expression of the transcription factors. 4 major transcription factors have been identified till now. The OCT3 or 4, in some cases SOX2, C, MIC and KLF4, out of which SOX2 and OCT4 function by activating NANOG and other transcription factors that establish pluripotency and block differentiation. While C-MYC opens up chromatin and makes the genes accessible to SOX2, OCT4 and NANOG, while KLF4 prevents cell death of these cells. So these regulatory transcription factors are essential to maintain the uncommitted stem cell-like state and functional pluripotency of the inner cell mass, thereby enabling the inner cell mass cells to give rise to the epiblast and all associated derived cell types. And at the same time, they help in repressing the genes which are involved in differentiation, thereby maintaining the pluripotent state of these cells. How are embryonic stem cells cultured? Embryonic stem cells were originally cultured by growing them on a layer of mouse fibroblasts, which are called the feeder cells, as can be seen here in the petri dish also. This culture is done in the presence of bovine serum and other growth factors. So in these petri dishes, the cultured inner cell mass cells are kept on a layer of irradiated mouse fibroblast cells, which are known as the feeder cells, plus the bovine serum or other growth factors are added. while the addition of the cytokine leukemia inhibiting factor, that is LIF, to this serum containing medium allows the mouse embryonic stem cells to proliferate even in the absence of these fibroblast feeder cells. Now, if there is some addition of growth factors like fibroblast growth factor FGF or platelet-derived growth factor PDGF in the culture medium, then the embryonic stem cells are able to differentiate into functional glial cells, as has been seen experimentally. If retinoic acid is added to the medium, it leads to the differentiation into functional neurons. So that means addition of different culture factors or different growth factors to the culture medium allows the embryonic stem cells to form different type of cells by differentiating different. So what are the applications of embryonic stem cells? The first major application is in the field of regenerative medicine or transplantation therapy. Patients can have their diseased organs regrown and replaced using their own stem cells. The embryonic stem cells provide an unlimited amount of tissue for transplantation therapies, and it helps in treating a wide range of degenerative diseases. Embryonic stem cells can be made to differentiate into insulin-producing cells under the condition of diabetes or dopaminergic neurons, as in case of Parkinson's disease, also in treatment of Alzheimer's disease or liver cirrhosis with different culture conditions in which the embryonic stem cells can be made to differentiate into different cell types required to treat different diseases. Second major application is drug testing and gene therapy. Embryonic stem cells can support basic research on the differentiation and function of human tissues by providing material for testing that may improve the safety and efficacy of human drugs. In addition, the embryonic stem cells can be genetically manipulated for gene therapy. There are also some risks or limitations associated with using embryonic stem cells. The first and the foremost being that human diseases involve cells that have a long history of differentiation events and are often multigenic, that is, they are caused by the interplay of many genes. So just using the embryonic stem cells to form the cells of the adult organs may find certain limitations. The second is the risk associated with immune compatibility. There is always a risk of immune rejection by the patients who receive transplanted embryonic stem cells as a part of a treatment. There are again some social and ethical issues since obtaining embryonic stem cells involves human blastocysts or the human embryos, and it is believed that they die during isolation of these embryonic stem cells. So, an alternative to using embryonic stem cells can be that adult stem cells can be used, and second is that induced pluripotent stem cells can be used. So now let us discuss these two. The adult stem cells are in fact multipotent in nature. They are present in numerous adult organs, for example, some cells of the epidermis, some in the brain, in muscle, in teeth, in gut, in lung, or the hematopoietic stem cells are multipotent in nature. However, obtaining these adult stem cells is little difficult, and they create a restricted array of cell types even if grown in under cultural conditions. And they bear a finite number of generations only, and for self-renewal, the limited renewal of adult stem cells may contribute to their aging. So the best alternative for embryonic stem cells is the induced pluripotent stem cells, IPSC, which even are able to overcome the limitation of immune incompatibility associated with the embryonic stem cells. Like embryonic stem cells, the induced pluripotent stem cells are also pluripotent in nature, as their name says. And how are they produced? They are produced by genetic reprogramming of adult somatic cells by inserting activated genes which are responsible for pluripotency. As we discussed, there are four major genes, OC3 or 4, SOX2, CMYC, and KLF4, which are responsible for pluripotency in the embryonic stem cells. So if those four genes are taken up and induced into the adult somatic cells, then they can also be induced to become pluripotent. And this is what Takahashi and Yamanaka of Kyoto University did in 2006. They experimentally demonstrated that by inserting the activated copies of 4 genes of 3 or 4 SOX2, CMYC, and KLF4, which encode critical transcription factors necessary for pluripotency, into differentiated mouse fibroblasts. So those mouse fibroblasts were able to de-differentiate into inner cell mass-like cells. That is, they were able to form pluripotent cells, and thus they were given the name induced pluripotent stem cells, IPS. Like embryonic stem cells, iPS cell lines can be propagated indefinitely, and whether in culture or in a teratoma, they can form cell types representative of all 3 germ. By 2012, there were some modifications done in the culture techniques which made it possible for the gene expression of mouse-induced pluripotent stem cells to become nearly identical to that of mouse embryonic stem cells. So the entire mouse embryos could be generated from a single induced pluripotent stem cells, which shows complete pluripotence. Although these cells are functionally pluripotent, they are best at generating the cell types of the organ only from which the parent somatic cell that has been taken was originated. So this is a limitation of these induced pluripotent stem cells. And what are the merits of induced pluripotent stem cells over embryonic stem cells? The major merit is that these transplanting cells, which are derived from the mouse-induced pluripotent stem cells back into the same donor mouse, does not elicit immune rejection. So there is no problem of immune incompatibility seen, though they are cost intensive and they show a low rate of production. Also, in this case, there are no embryos involved, so there is no ethical issues involved, hopefully, with these stem cells. Now, what are the applications of induced pluripotent stem cells in human development and disease? Using these induced pluripotent stem cells, the medical researchers are able to experiment on diseased human tissues while avoiding the complications which are introduced by using human embryonic stem cells. Using patient-specific iPSC-derived pluripotent progenitor cells in cell transplants without the complications of immune rejection. And these induced pluripotent stem cells have found applications in disease modeling, that is making patient-specific induced pluripotent stem cells for studying disease pathology and combining gene therapy with patient-specific induced pluripotent stem cells so as to treat a particular disease. They also find applications in regenerative studies where they are widely utilized for the generation of tissue-specific cells to be transplanted to patients in various injuries or degenerative diseases. In drug discovery, in screening of small molecules, in toxicity assessment, and in testing studies which utilize differentiated cells, here the induced pluripotent stem cells directly derived from the patient can be used in certain experiments. So this is all about the lecture, and today we have discussed 3 small topics, that is in vitro fertilization, amniocentesis, and also the embryonic stem cell. And also we have studied various variants of in vitro fertilization and the embryonic stem cells. Also we have discussed the merits, demerits, and limitations of each of these. So let us summarize this lecture in a snapshot. The IVF, which stands for in vitro fertilization, which is an assisted reproductive technology or ART, involves hormonally controlling a woman's ovulatory process for retrieving ovum from the woman's ovary and fertilizing them with the sperm in a culture medium, which is kept in a petri dish, and transferring the resulting embryos back to her uterus 2 to 6 days later. Now, eggs are collected directly from the follicles or after ovulation. The sperms are collected and concentrated from the male partner. Both the collected egg and sperm are mixed in a petri dish, allowing fertilization to occur. The fertilized zygote is removed from the petri dish and surgically implanted into the mother's uterus to develop non. Various variants of IVF are ICSI, GIFT, ZIFT, and FROST. The amniocentesis is a prenatal diagnostic test for detection of genetic disorders and infections using fetal DNA from the cells collected from the amniotic fluid, which is carried out between 14 to 18 weeks of pregnancy. A small volume of amniotic fluid, around 20 to 30 ml, containing fetal cells is aspirated with an ultrasound guided needle. Fetal cells are grown in the culture medium and examined for chromosomal abnormalities. Amniocentesis is applicable in fetal sex determination, in genetic testing, in fetal lung testing, diagnosis of fetal infection, polyhydramnios, and paternity testing. The embryonic stem cells are derived from the inner cell mass of an early embryo, primordial germ cells, or in vitro fertilized embryos. Expression of the transcription factors, that is OCT3, 4, SOX2, 6-CMYC, and KLF4, is essential for the transient pluripotency of the inner cell mass. The originally derived embryonic stem cells are grown on a layer of feeder cells in the presence of bovine syndrome and other growth factors or leukemia inhibitory factor, that is LIF. Embryonic stem cells have applications in regenerative medicine, transplantation therapy, drug testing, and also in gene therapy. However, immune incompatibility and ethical issues involving death of the embryos pose limitation to their use. Hence, induced pluripotent stem cells, or the iPSCs, which are produced by genetic reprogramming of adult somatic cells by inserting activated genes responsible for pluripotency, can be used as an alternative for embryonic stem cells. The transplanting cells, which are derived from mouse, induce pluripotent stem cells back into the same donor mouse, does not elicit immune rejection. And these cells, just like embryonic stem cells, also have applications in disease modeling, regenerative studies, and also in drug discovery. These are some of the books which have been referred for preparation of content of this lecture. Hope you had a good time listening to me. Thank you. Wish you a very happy learning.